Welcome to SB class by Angel. Okay, welcome to SB class. I'm going to talk about some topics that I saw. So we're going to start. The first one is the inheritance. The inheritance is the process by which the traits of organisms are passed to their offsprings. What is the physical basics of inheritance? Genes are sequences of nucleotides at specific locations on chromosomes. Inheritance is the process by which the characteristics of individuals are passed to their offsprings. A gene is a unit of heredity that encodes information needed to produce proteins, cells, and entire organisms. Genes comprise segments of DNA ranging from a few hundred to many thousands of nucleotides in length. The location of a gene on a chromosome is called its locus. Right now, I'm going to explain something about the chromosome. What are chromosomes? Are from cells package their DNA. His DNA protein complex is called a chromatid. Wherever the mass of proteins and nucleic acids is nearly equal, within the cell chromatin usually falls into characteristic formations called chromosomes. Each chromosome contains a single double-strand piece of DNA along with the aforementioned packaging proteins. How much chromosomes are needed to create an organism? 22 of these pairs, called autosomes, look the same in both male and female. The 23rd part is the sex chromosome. What are nucleotides? Nucleotides are organisms compounds that consist of three joint structures: a nitrogen base, a sugar, and a phosphate group. The most common nucleotides can be divided into two groups: primers and premixes. Based on the structures of the nitrogen base, the joint sugar is either ribose or deoxyribose. Nucleotides are the structures of units of RNA and DNA. They also serve as important cofactors in cellular signaling and metabolism. Their cofactors include CoA, flavin adenine dinucleotide, flavin mononucleotide, adenosine triphosphate, and nicotinamide adenine dinucleotide phosphate. Now I'm going to talk inheritance again. Homologous chromosomes carry the same kind of genes for their characteristics. Genes for the same characteristics are found at the same on both homologous chromosomes. Alternative versions of genes found at the same gene locus are called alleles. If both homologous chromosomes carry the same allele gene form at a given gene locus, the organism is homozygote at that locus. If two homologous chromosomes carry different alleles at a given locus, the organism is heterozygous at that locus. Now I'm going to explain about the Punnett square. The Punnett square was created by Punnett and phenotype. In this case, the plant that we used was the pea plant. So our phenotype is this one. In this part is the trait. You can see the seed shape, the seed color, the pod shape, the pod color, the flower color, and the flower location. The dominant form in the seed shape is smooth. In the seed color is green. in the pod shape is inflated, in the pod color is green, in the flower color is purple, and the flower location at leaf junctions. And the recessive form in seed shape is wrinkle, in seed color is green, in pod shape is constructed, in pod color is yellow, in flower color is white, and in flower location is at tips of branches. Genotype. Our genotype is homozygous dominant, homozygous recessive, or heterozygous. This can be measured as fraction, as a ratio, as a percentage, or just the number. Don't forget that the homozygous dominant and the homozygous recessive are equal, and the heterozygous is the different. In this case, in this exercise, I'm gonna use the capital X in the seed shape like smooth, and the lowercase x as wrinkle, and the P capital will be green, yellow, and the P lowercase will be green. The F0 will be X capital, XX capital, XX lowercase, PP capital, and PP lowercase. In the F1, what we make is an exchange. So we make this with X capital with P, X capital with P lowercase, X lowercase with P capital, and X lowercase with P lowercase. So in the F2, we will have this by making this the F1 procedure. So with this, I can make our Punnett square, and in the Punnett square, I can know the phenotype and the genotype. With the F2 pattern, I must make the Punnett square. So that pattern, I use it in the first line and in this part. So how works our Punnett square? Okay, our Punnett square works like this. XP plus XP will be XXPP. XP plus X and lowercase p will be XXP lowercase p. So what we make is to combine those, and always the dominant will be at the first. If you look, the P is the final one, will never be in here. So can be in here because if it is X, but always at the final of the pattern. So with this, I can know our genotype and our phenotype. The homozygous dominant that we have here is this one, that is the only one in all capital letters. And the dominant recessive is this one because it's the only one that have lowercase. So with this, I can make predictions of generations.